This is the pre-lab lecture for our selective and differential media, which encompasses exercises 4-1, 4-3, 4-4, and 4-5. First, background about selective and differential media. To be selective means that the media contains ingredients that will promote the growth of certain microbes and inhibit the growth of others. That's what it means to be selective. We choose a media that will select for the growth of some microbes and inhibit the growth of others. So the first example we have of that is our PEA, which stands for Phenol, Ethyl, Alcohol Agar. That alcohol inhibits the growth of gram-negatives and allows gram-positives to grow. That happens because gram-positives have that very thick peptidoglycan layer, and the alcohol has a hard time getting into the cell. Gram-negative cells have a very thin peptidoglycan layer. The alcohol passes through that wall and damages the DNA. Because gram-positives can grow and gram-negatives may not be completely killed, but they will be inhibited. They will not grow well. PEA is called selective for the growth of gram-positive. So if we had growth on a phenyl ethyl alcohol plate and on a plain media like tryptase soy agar, if we had good growth on both, we could deduce that the organism on the PEA plate is gram-positive. If we streaked an organism on PEA and TSA, and it grew really great on the TSA, the non-selective media, the media that supports the growth of everything, but it grew poorly on the PEA, we would deduce that that organism is gram-negative because PEA inhibited. The second selective media we have is MSA, and that stands for mannitol salt agar. This is exercise 4-3. It has a very high salt concentration of 7.5% sodium chloride. Normal saline is around 0.85%. So this is nearly tenfold what the salt content is in our cells. Because of that high salt content, mannitol salt agar is selective for the growth of sodium chloride tolerant microbes, and that's primarily Staphylococcus. There will be a few other gram-positive microbes that can tolerate this media, but really the Staphylococcus and maybe Bacillus are the only ones that can grow well. Other organisms, if we streak it on this mannitol salt agar, grow extremely poorly or not at all. Exercise 4-4 is the MAC, or Mcconkey Agar. It has bile salts and crystal violet, and both of those inhibit the growth of gram-positive. We saw crystal violet in our gram stain, and that sticks to the peptidoglycan as the cell walls are being made, when the bacteria are trying to grow on that agar surface. And when the crystal violet is sticking to growing cells, it prevents those layers of peptidoglycan from building up, and the gram-positive cells cannot build their cell walls. They'll grow poorly or not at all. The gram-negative microbes only have one layer of peptidoglycan, so the crystal violet sticks to that. The cell wall is already made. It does not inhibit the gram-negatives. So the Mcconkey agar is selective for the growth of gram-negatives and against the growth of gram-positives. The bile salts also inhibit the gram-positives, and they don't bother the gram-negatives. Many of the gram-negative organisms that are on our unknown list are found in the digestive tract. Bile salts are a chemical in the digestive tract. So gram negatives are often resistant to that, and the gram positives cannot grow in their presence. And the last media for which we'll discuss selectivity is EMB. That stands for eosin methylene blue. Those are both dyes that again interfere with the cell wall layering in gram positives. So they are selective for the growth of the gram negatives, and it's selective against the growth of the gram positives. So we have 4 different media, and they are all selective. They will promote the growth of some microbes and inhibit the growth of others. 3 of these media are also differential. And to be differential means of the organisms that grow well, there are color indicators that will detect different metabolic activities. So organisms that grow poorly on the media, they were selected against. We can't really determine anything else about them other than the media inhibited them. And that may tell us something about whether they're gram positive or gram negative, but it doesn't tell us anything about their metabolic activities. You have to have good growth in order to say anything about their metabolic or different metabolic activities. The PEA agar, exercise 4-1, is only selective. However, the other 3 media are also differential in addition to being selective. So exercise 4-3, the mannitol salt agar is selective for the salt-tolerant microbes, and then the mannitol, the M portion of the MSA, is a sugar that can be fermented. So this media will tell the difference between organisms that can ferment mannitol versus the ones that cannot. Again, the bacteria have to grow well. If there's no growth, we cannot say if it ferments mannitol or if it does not. A lack of growth tells us the bacteria is not salt tolerant, and that's all it tells us. It has to be alive and growing well to determine if it ferments mannitol. We can determine that fermentation because this agar contains phenol red, that is a pH indicator. It is red to pink if the pH rises above 6.8, and it is yellow if the pH drops below that. And so if on the agar, which initially is red, there is growth and the growth leaves the agar red, then that organism cannot ferment mannitol. However, if there is growth and that growth generates acid because of fermentation of mannitol, the media will turn yellow. So on our slides, on our images, our still images of the media that have been inoculated with the bacteria, there will be a positive control where there will be growth and the agar turns yellow because fermentation of mannitol makes an acid that lowers the pH. The media turns yellow. We will also see bacteria that can grow on mannitol salt, so it is salt tolerant, but the agar will stay red. And if we have good growth and the plate stays red, we know that it is salt tolerant and we know that it cannot ferment the mannitol. The Mcconkey agar had the bile salts and the crystal violet that inhibit gram positives, so it is selective for the growth of gram negatives. And of those gram negatives that grow well on the media, we can determine whether or not they ferment lactose. So MSA had mannitol as a sugar for bacteria to ferment. The Mcconkey agar has lactose. Now, if the bacteria do not grow, or they grow very poorly, we cannot determine whether or not they ferment lactose. All we can say if a bacteria grows poorly is that it is not gram negative. That's it. This media is useful for looking at coliform bacteria. That coli we saw with E. coli, and all coliform bacteria are found in guts of animals. That's why they are coliforms, from the colon. All coliform bacteria have these three characteristics. They are gram-negative, they are rods or bacillus-shaped, and they ferment lactose. There are lots of gram-negative rods that don't ferment lactose. There are plenty of gram-negatives that ferment lactose and aren't rods, but to be a coliform must have all three of those features. This media will change colors because it is differential, right? To be differential says of the bacteria that grow, we can see a difference, and that difference is because of a metabolic activity. For Mac, that metabolic activity is fermentation of lactose. So when bacteria are streaked or inoculated onto Mcconkey agar, we can have dark pink. That means that acid was made from fermenting the lactose. It causes a color change. Or we could have no color change, and that means the lactose was not fermented. So if the bacteria grow and it turns pink, it is gram-negative because it grew. It is a lactose fermenter because it changed colors. If bacteria are inoculated and they grow well, but there is no color change, it's still gram-negative because of the good growth, but it does not ferment lactose. If we were to inoculate a third quadrant with a bacterium and we saw no growth, or we saw very poor growth, all we can say is that it's probably gram-positive. We have no idea whether or not it can ferment lactose because it has to be alive in order for us to determine that activity. The fourth plate is the EMB, the eosin methylene blue agar. That was also selective for gram-negatives because the dyes eosin and methylene blue inhibit gram-positives. So of the gram-negatives that grow well, we can tell the difference as to whether they ferment lactose and or sucrose. So only the gram-negatives can grow. If we inoculate a plate, if the color of the agar turns dark purple, it ferments the sugars lactose and or sucrose with a lot of acid being made. If the growth turns a shiny green, it's gram-negative because it grew. That color change also indicates it ferments lactose or sucrose. And if it grows well but has no color change, still gram-negative, but it doesn't ferment lactose or sucrose. And if we had a fourth quadrant with inoculation that did not grow, all we could say would be that that microbe is gram-positive. Without good growth, we cannot determine whether or not it ferments. That's the principle. You can also refer, you should also refer to the theory in your lab notebook, the Leboff-Pierce manual for exercises 4-1, which is the phenol ethyl alcohol agar, 4-3, which is mannitol salt agar, 4-4, which is the Mcconkey agar, and 4-5, which is the eosin methylene blue agar. You will be provided with still images of controls on all four of these media and tryptase soy agar. That is the control agar. Everything should grow well on that. So in each of the images, you need to compare the growth on the exercise plate to the growth on the tryptase soy agar plate. That is how you will determine if the microbe grew well or grew poorly on the selective and or differential media. You compare it to the growth on a non-selective, non-differential media. You will also have the still images of your unknown on each of the 4 selective and or differential media and on a trypticase soy agar plate. So when you determine whether or not your unknown grew well, compare it to the trypticase soy agar plate, and then you can compare, once you know the growth is well on a plate, if it has the activity you're looking for or not. So if your unknown grew really well on the PEA plate, you know that it is gram-positive. And then it may or may not grow well on any of the others. It has to really love salt to grow on MSA. And if it is a gram-positive, it should grow poorly on the MAC and the EMB. If you have a gram-negative microbe, it should grow poorly on the PEA compared to the TSA. It should grow very poorly or not at all on the mannitol salt agar, because gram-negatives do not tolerate salt very well. They have that thin cell wall. But your gram-negative should have really nice growth on the Mcconkey and the EMB agar. Then you need to look at that good growth and determine if your microbe did or did not ferment the sugars, and that will be based on their color changes.